This conference will now be recorded.
Very good evening from Biosetup Lifesciences. We are sincerely apologize for the unusual delay in starting today's webinar. We encountered unforeseen technical difficulties that took longer than expected to resolve. And thank you for your patience and understanding. We are now ready to begin shortly. Welcome everyone. Good evening, ladies and gentlemen. I, Navamita, extend a warm welcome to all the esteemed participants joining us today. We're delighted to have you all today for this webinar on Angiostrongylus vasorum, canine lungworm, etiology and importance of a good diagnostic strategy, organized by Biosetup Life Science. Let me enlighten you all about the Biosetup Life Sciences. That Biosetup Life Sciences founded by Mr. Orghodip Das. So it is a hub for scientific research and learning approved by ISO 9001 2015 certification. We have multidisciplinary team of faculty and associates. We are continuously working for the career upgradation of the students. We have successfully organized many workshops, courses, career counseling, competitive exam preparation like CSIR, UGC NET, IIT JAM, CUET PG, GAT-B, IIS, SC exams and DBT exams. We provide support to the aspiring student in the field of life science. So, ladies and gentlemen, it is my distinct honor and privilege into the introduce our esteemed keynote speaker, Dr. Katarina Novakovic, for today's webinar session. Dr. Katarina Novakovic obtained her degree Doctor of Veterinary Medicine from University of Belgrade, Serbia. She has experience in the field of veterinary laboratory diagnostics for 16 years now, and presently she is working in Smart Lab as senior laboratory consultant. She is proficient in hematology, clinical biochemistry, and parasitology, and has conducted analysis on a wide range of species, including wild and rare animals. She was lucky to collect samples from alligators, eagles, turtles, alpacas, bearded dragon, and many other exotic animals. In addition to her laboratory work, Dr. Katrina played a key role in implementing ISO IEC 1025 2017 standards and reliability of veterinary laboratory practices. Dr. Katrina is deeply passionate about animal welfare, public health, and human education. She advocates for replacing harmful animal use in education with non animal models and promotes sustainable practices through permaculture. Her commitment to these cases reflects her dedication to advancing both veterinary science and ethical practices. So before we commence our today's presentation, we have a short announcement of a little housekeeping. So before we start the meeting, please don't share your audio and video during the session. If you have any question, write it down into the chat box and we will take your question at the end of the lecture. So, without taking too much time, I would like to invite Dr. Katrina Novakovic. The floor is open to you, ma'am. You could commence your presentation. Thank you.
Thank you, Navamita. Just to be sure that you can hear me, is it okay?
Yeah, it is completely okay.
Okay, good, great. No, no, this is my first webinar, so yeah, I usually talk in live, so yeah, this is new for me. But I'm really glad to have this kind of experience with all you students and yeah, people from all around the globe. So as you can see, this is one of my passion and one of my yeah, one of my things that I really like, parasitology. And when we talk about topic, this was interesting because of the diagnostics, because of how to manage, how to find out is it this parasite or not. And I would like through this topic just to talk about Angiostrongylus vasorum, but also to talk about parasitology and strategy in parasitology in general, because it is important, and you can find sometimes a problem to choose how to do, how to approach, etc. And I hope that um, uh, I will do a little bit to clarify and to help you in a future to do some diagnostics in parasitology. So as you can see, Angiostrongylus vasorum is canine lungworm, but you can find lots of different names of this parasite. This is picture of larva first stage, and it is important to see how it looks like, because it's going to be important for diagnostics afterwards. So this is really nice picture, and just observe tail in the center. Angiostrongylus vasorum is nematode parasite, lives in pulmonary arterial tree of dogs and foxes. And foxes are really important because they are spreading, and they are like reservoirs for these parasites. Firstly, we could find in Europe, but also I find lots of data about these parasites found in Uganda, Turkey, Australia, South America, and nowadays in North America. So we can say that on every continent we have these parasites, at least in one country. This parasite is 2.5 centimeter adult form, lives in pulmonary arteries in dogs and foxes, as I said. Larva stage 1, it's important. You saw the picture, really nice one, but we are going to see how can we find this larva once the first stage. So that larva enters airways. Dog and cats, they are coughing. They are swallowing. So that larva is going through airways to the stomach, and then in feces. Then from the feces, usually snails and slugs, um, larvae invade snails and slugs, and they are intermediate hosts. Uh, Maybe you for sure you know that the difference between intermediate hosts and paratenic host, but just to clarify, intermediate host is a host for parasites where parts of development occur. So usually from larva first stage to larva third stage, in intermediate hosts that development occur. Paratenic hosts, they are just bringing mechanically larva, and no development occur. So larva 3 are third stage are in snails or slugs. They are again eaten by dogs or foxes, and third stage larvae penetrate from gut wall and migrate to mesenteric lymph nodes. Then they develop from the third stage to fifth stage, and this is a stage that is actually one step to adult. Fifth stage larva is then going to the pulmonary arteries through blood, and these all cycle from larva uh, last 40 days. So Angiostrongylus vasorum is called also French heartworm because the first reported case that we knew was in France in 19th century. But still then, as I said, we can say that every continent, at least in one country, has this worm. This is a map of Europe, and you can see my country, the green one, Serbia. On the west part of Europe, that the worm started, and you can say by number of cases are the most in France, England, Spain, but also in Central Europe. But you can see that um, during the time, because of lots of movements nowadays, that parasite spreads, and we could find on the northern part of Serbia, and then in the southern part of Europe, Greece and Turkey. So yeah, so it's now we can say that whole Europe is in this agenda. Important part of diagnostics is also know the epizootiology, of course, but also to know clinical features. And this is a tricky part because you can find almost asymptomatic to severe disseminated intravascular coagulopathy due to immunomediated hemolytic anemia. But also the most common are cough, exercise intolerance, weight loss, hemoptysis, coughing of blood, subcutaneous edema to heart failure and perihemial lung disease, various difficulty, respiratory difficulties, diarrhea. And also what is important to know that usually if dogs are eating snail and slugs, they are usually ingesting lots of other parasites. So you can find competitive parasitosis, intestinal and lung parasitosis, and more severe clinical signs are, like I said, thrombocytopenia or this disseminated intravascular coagulopathy due to immunomediated hemolytic anemia. And this is obvious, this last one, IMHA. It's really obvious because all blood parasite, and this is blood parasite, actually, it is in lung, but because of the clinical signs, but actually you could see that this worm is situated in arteries. So everything that is living and situated in blood can cause immunomediated hemolytic anemia. Actually, lots of foreign antigens in blood vessels can cause severe immunological defense, and then the body can attack their own erythrocytes, and this is pathophysiology of this kind of disease. Important thing to know about this is that your approach to diagnostics should start when you enter, for instance, if it's shelter or group animals of you could hear that in area you can have similar signs, clinical signs, but not same, because dogs and animals and we are all different. So you can expect that they're not exact, and you can have various, like I said, various clinical signs, and you can have really tricky, not obvious ones. So you should start with, my suggestion is always to start with hematology. This is the first step always, and you should do that. And you can have anemia and low thrombocytes in this hematology picture, and also you can have high leukocytes going along with that. if you do thoracic radiology, X-rays or ultrasound, it is important that you will have a picture of enlarged heart, probably, and different um, uh, pulmonary differences in X-ray, and this is important because other other parasites are not making this kind of pathology. The main diagnostics, and I can, because it's really easy to do Berman technique, I'm suggesting just for fun to just try to make this technique, to learn to make this technique, and then to use it whenever you can. You really don't need to have a high-tech laboratory for this, and you're going to see what is needed. Really, nothing special, but it's something that is highly used even now. Serology is always a good approach if you have it, antibodies or antigens, or you can do from blood. And luckily, we can have, because it's not it's actually blood parasite. We can do also along with serology PCR and confirm diagnosis. And what is not so new thing to know, but really important one. You can see one special peak, beta 3 peak in serum protein electrophoresis. And this is also one thing that there is used to do diagnostics. So as you can see, and other parasitology diagnostics are similar to this. You can approach from different sides, and in practice, we are mostly using Berman technique and serology. Uh, so Berman technique is really simple. You need one glass, plastic one. You need one cloth, but should be with a big holes like gauze or something like that. You have in all veterinary clinics, and warm water, not cold, not, well, warm, but like body warm, like 30 to 40 degrees, not hot, and that's it. Um, to fix somehow, this is one suggestion, to fix somehow goes to the wall, and you put water like this at one point, put a number or name of the animal, and also the timing is important. You should put at least 8 hours to sit there in the water. Um, I usually do today for tomorrow, and this is working well. Um, uh, the thing is that in warm water, larva, the first stage larva, we could see at the beginning, the first stage larva is going out from feces, and you should use like 3 small spoons of feces as a sample. So larva from feces is going out, and because it's not, it cannot swim, it's just falling down. And after 8 hours or more, you can collect from the bottom, and it should be glass like this that is narrowing down. So the bottom part is small, so you can pipette it, and you usually see larvae. But all larvae, they are really similar, and you should be aware that other larvae from different kind of metastrongylid, that this parasite is metastrongylid, they can be really, really similar. This is larva, Angiostrongylus vasorum larva. So when you pipette from the previous slide, so you pipette it, you put on a glass slide. When you find larva and it's moving, you use Lugol iodine solution, from gram stain, for instance, or whatever you have. And this solution is killing larva. Larva is sitting like this, and you can observe cephalic bottom on the bottom bottom right side. It's cephalic bottom. Sorry, and but most important thing, you can see the tail. There is a really S-shaped body, but tail is really distinct. And this is almost as identical as a similar parasite to cats, Erolostrongylus abstrusus, but no other parasites can have this. So just remember this picture, because it is crucial for diagnostics. If not, if you're not sure, if you're not skilled, but as you can see, you don't need to be so skilled. Some skill you need to have. If you're not sure, you should do serology or PCR, just to be sure if it's this parasite. Why I was stressing this tail shape, mostly tail shape, you could observe, because other lungworms and heartworms or other larvae you can find by Berman technique or other technique. You can have in your feces sample, or like Dirofilaria imitis in a blood sample. So just to be sure. Crenosoma vulpis, Philoroides osleri, Strongyloides tercularis. You can find this larva in Berman technique. First two, they are lungworms. Strongyloides tercularis is usually asymptomatic, but you can really often find on Berman technique. So one more time, observe this tail, and if you find this kind of tail, you know that there are not three of these. Dirofilaria imitis is a heartworm. I'm not sure if you have it in your region. There are other heartworms in dogs, of course, but it's easy to prove to prove by other techniques like knot technique, Dirofilaria, or other kind of techniques or serology. But we have actually two heartworms, Dirofilaria imitis and Angiostrongylus vasorum, our today parasite. So this is the main thing to learn. Yeah, you can have you can use these kind of drugs, therapeutics. Also, in a region that you have really common these parasites, you can use it, and you should use it as a prophylaxis, um, febendazole or levamisole, um, or you can use it as a therapy. And note that you should not must not use levamisole and pyrantel at the same time. So don't just use two different antiparasitic at the same time if you're not sure if they have and they're using the same receptors and agonist or antagonist. One more parasite that you are for sure aware of, Angiostrongylus cantonensis. I know for sure that in Asia and Africa you have it. It's a lungworm of rats and humans, and it is a zoonosis. But you can find, and it is proof that you can find in a canine, in dogs, but in a nervous system. So it's different way, different diagnostic, and different etiology. But yeah, be aware that Angiostrongylus cantonensis you can find in dogs too. I would use serology in this kind of diagnostics um, to be sure that we have differential diagnosis. Also, as I said in the intro on a poster, it is important to know that different kind of diagnostics, and I will go back to this slide. So you can see clinical features, and it's really long list of differential diagnoses. It can be cardiovascular, allergic, trauma, neoplasia, inflammatory diseases, different parasites, as we said. So my suggestion would be to just observe and be sure that you used different kind of diagnostics. It's always good to start, as I said, with hematology, and in this case, just to put differential diagnosis on a paper and try to eliminate one by one. And it is tricky, but yeah, parasitology is fun, and you can find that it is actually easy to observe. So this would be the short part of my presentation, and I would like to ask you for questions, and we can just start to talk about it if you are okay with that. How to calculate well, this is a good question. I will read about dosage here. Um, I would, because you can do Berman, and then you can count larva, but it's not always the good strategy, and it is important to know that you will need. I would do that. You will need to do again Berman after a first cycle of therapy, and I will do, if you're not sure how strong is infection, I would start with ivermectin. You need to use 0.2 milligram per kilogram, and I will do or febendazole twice daily for 2 to 3 weeks. So there are different strategies, and after that, I will do Berman 5 days after the last therapy. I will do again, and as in Dirofilaria imitis strategy, I will do the same. I will do 3 Berman in a few days or 1 week apart, and then if I have a 3 negative Berman, I will say that this is good. Of course, you always watch for clinical signs and do hematology all the time, because usually you will have other clinical signs than just coughing. The patient version. Okay, yeah. Sorry for this. In the intro, it was my typing mistake. It was not about ethnicity. It's etiology. So that was some mistake I made, probably with the autocorrect. About humans, I know that for sure, and and I think that you're probably better know about Asia and the other Angiostrongylus cantonensis. But I would say that this zoonosis is probably most severe in countries and in countries that they're sentinel. They were not in contact with that parasites. So I would say that in Europe, and we have cases of Angiostrongylus cantonensis in humans, I think they are more severe because usually it's a last idea to go in that direction, and probably in Asia you are better equipped and better educated in that part, and also you have also immunology status. But I'm not in this kind of diagnostics. I'm not familiar. Is there any another techniques which we can use for diagnostics? Very good question. As I said, and I will show you on my screen. Sorry. I will do all this or some of this. I will always start with Berman. And also, you need to know that none technique is 100% sensitive and specific. It can be specific, but it cannot be sensitive 100%. So I would say that if your gut feeling is that there is a problem with this parasite, or if there is a problem with some of parasites, you should do Berman technique with, for instance, three feces in a row or that kind of stuff, and also do other like serology or PCR. So you should always start with Berman because it's easy and it's really good, really specific and really sensitive. And you know that specific is when it's like highly specific for that species. It's not going to show you false positive, and if it's sensitive, they're not showing a false negative. So you should start with Berman and hematology, and then you should spread with other techniques and other approaches like radiography in one hand, and the other one is serology. And I will always suggest to do serology because for serology you have enough time. It's 40 days, like I said. In 40 days, antibodies are present. Then you need 7 days from the beginning of the infection to show first antibodies, first titer. In 14 days, you have full titer usually of antibodies. So in 40 days, you will for sure have antibodies in the system, and you should do this. You should approach from that part. So I will say Berman and serology is a best approach. Okay, let me see. Sorry. Does it take full life cycle to infect? Yes, this is also a good question. So I said in full detail, larva, first stage larva to third stage larva, because it's um, it's really it's really nice to see on this example how parasitology and parasite strategy is really beautiful, because parasites are using bodies. Their strategy is not to kill a body. So intermediate hosts are used for developing of larvae. So host like dog, for instance, as a final host, they need um, third stage larva to infect. That means that they cannot auto-infect. In other hand, first stage larva it's just enough to spread the disease, but not to infect host again and again, because in that case, host would be dead in a few days or a few weeks because it would be infected really, really too much. So host needs to ingest snail or slug with third stage larva, and then like I said, add the development is going to happen. And after fifth stage larva, we can have adults in a blood system, and this is infection. Okay, Berman technique. You said it's a new technique. Okay, I will do for sure. I love that technique because it's really simple one. So you collect three feces from 3 days or one day. It depends that if it's their diarrhea present or not. So you collect three feces. You can do with one, but it's better two or three. Then you take 3 spoons of the feces and put in gauze like this. So it's like gauze that they're using you to cover a wound or that kind of stuff. So when you put it in a gauze, you just fix it to the glass and you put in a warm water. I usually just you can use tap water. You don't need to use distilled water or saline. You can use saline, but tap water is totally okay. But you can use saline, but it needs to be warm. Why? Larva from feces is going out from the feces to the warm water. And because of gravity, they cannot swim. Larvae cannot swim. Because of gravity, they're going to fall down. And you need at least 8 hours for all larvae from the feces to go out. Then they are going to fall down in a glass, in a bottom glass, and then you remove feces and you pipette from the bottom. You will have something like a small sediment, really small sediment. You pipette sediment from the bottom, put on the glass, microscopic glass, and then you observe. If you find larva, and it's really easy to find larva, then you do second step. You put iodine, you put Lugol solution in order to kill larva and in order to observe larva and morphology of larva here, to observe tail and cephalic pattern. If there any initial symptoms observed during incubation period in host body? In incubation period, no. Maybe they can be diarrhea because of movement larva through gut wall. So yeah, some digestive problems, some diarrhea or that kind of abdominal pain, but not severe. Really mild one. And then maybe some during that movement large lymph nodes mesenterial, but you cannot see that. It's really something that is happening in the abdomen. And then the first symptoms you can actually see is when larva 5-stage larva is moving to vascular system in a blood vessel, then you can see first symptoms. And as you can see, while that larva and adult form is moving and living in a position, you can have different kind of symptoms. what the minimum time of incubation period for showing the symptoms, it really depends on individually, and depends on number of larvae. But I would say that in 1 month you can have adult form. This is the minimum. And yeah, the diarrhea can be first time. You can see and yeah, observe. And I would like to invite you, if you have any other questions or any other ideas, and especially about laboratory diagnostic, I can help you, uh, mostly through LinkedIn. I can we can be in contact, and yeah, you can ask me whatever you need from this or similar topics. What is the best thing to do after completing? Okay, this is not for me. Can you just, is it the last question for me about PCR or? Okay, so first, Pranavi, what the best thing to do after completing BTech biotechnology? Well, I was studying and it's for after complete. Uh, well, I was studying veterinary medicine, so I'm not sure what is your your degree, what is your idea, what is thing you should do. But yeah, the laboratory diagnostics, especially if you can combine human and animals, or even better, One Health, human, animals, and environment, that would be my suggestion as the best thing to do. My field is a little bit tricky because I'm just working in clinical pathology for animals, and it's a lot of different kind of knowledge, but you are just in that field. So if I need to choose one more time, I think that I will do some One Health approach, and yeah, try um, to be in a field with human veterinary medicine and environment. Any control measures? Yeah, that is good question. For sure. For instance, in Switzerland, it's really in the center of Europe. They're suggesting to cover animals with antiparasitic every few months, because they know that it's really spreading a lot, and they are really in a great problem. So control measures should be antiparasitic drugs, depending of antiparasitic, but in a few months, and especially because it is proved that even that dogs don't even need to ingest snails or slugs. Sometimes just they can ingest grass after passing snail or slug in that grass or puddle. So it's really in areas that they are infected with this parasite. It's really mandatory to do some prophylaxis. What is the cure? Antiparasitic ivermectin, febendazole, or levamisole. Vaccine? No, as I know, no. I didn't read about vaccine. That's why Berman is important, because it's easy, it's cheap, but very sensitive and very specific. And through that technique you can also find other parasites. This is not for Angiostrongylus. You can find other larvae, other parasites, and really important one. And yeah, even like I said, even dog, they don't need to ingest snails. They need to ingest water or grass. They were in contact with the snails or slugs. I think that there was a one question about molecular biology techniques. So if I understand, you ask about diagnostic through molecular techniques. Yes, PCR is used, and you can do two different ways. You can use first Berman technique, collect larvae, and then these larvae do PCR. This is the first thing you can do, and also, luckily, because this parasite is living in arteries, you can collect blood, and from adult womb, from adult form, cells from that body are falling down in a blood. So you can do PCR from the blood, but also with PCR, you need to be aware that you need to have at least 300 DNA or RNA units. So if there is a small infestation of infection of this parasite, if immune system is working well and collecting these foreign cells, probably you can have a false negative. So with diagnostic in general, in parasitology and in general diagnostic, negative or positive are not always negative or positive. So be aware that it can be false negative and false positive. And like I said, false positive you can have if you're not observing larva in a good way, and false negative you can have if you don't have larva or anti gene, or yeah, for PCR. So just combine diagnostics. Flotation and sedimentation technique, really good question. Um, you can find on flotation, but this is really rare, and I'm not suggesting that. Sedimentation, nothing special. Yes, but you can have, but it's really, comparing to Berman, it's like You can find on Berman 10 or 15 larvae, and on flotations and imitation from the same sample, you can find 1 or 0. So you can find by chance on flotation and sedimentation, but this is not a technique that I like to use. Can we do treatment with survey effects on this? I am not familiar with that. No, I'm not. Really nice question. And yeah, I need to say one more thing about treatment. In heartworm diseases like Dirofilaria and Angiostrongylus vasorum, it's not difficult to, let's say, kill that parasite. We can do that with antiparasitic, but it is problematic if you kill it fast. You can have that parasite can clot and makes really severe pathology, and even we can kill animal. So slow killing techniques are preferable in treatment, especially of Dirofilaria imitis, but also in this case. So slow kill technique in some period of time, like 2 to 3 weeks, is better, especially if we don't have idea how many adult form we have. Does temperature have any effect on it? I'm not sure. This is also a nice question because we have in western part of Europe and the northern part, but I would say that this parasite actually don't have a problem with temperature, cold or not cold. It's using snail and mollusca, snail or slug. So I would say that especially because we know that they can they are present in Africa in that large snail, African snail. So I think that it's not a problem. Could you please put some more light on the morphological differences of parasitic samples being studied from intermediate and definite hosts respectively, or about the specific morphological differences? I don't have pictures, or I have just a larva, first stage larva. This is really nice question, but really difficult to obtain because larva 3 is present in a snail ingested. We really can, it's really difficult to catch it. And also larva fifth stage is present in lymph nodes, so it's really difficult to have it. You can find by accident or not accident during autopsy, for instance, but in a live animal, it's really difficult. So I'm stressing first stage larva because it is possible for us to observe it, have it, and then find it. So it is interesting, but really, really difficult to have it. And heartworm, it's really difficult to catch it in a live animal, and that's the reason why we are doing antigen serology or PCR. In the case of dirofilariosis, if we gave immediately the medication at full dose, cause of death host, so how should we treat? This is also a good question. There is a protocols, really, really nice protocols. Dirofilariosis is strategy for dirofilariosis is like this. Dirofilaria imitis is always going with bacteria, Wolbachia. So you start with doxycycline. tetracycline antibiotic. And after that, you start with slow kill protocol. In filariasis, it can last for 6 months to 1 year. Depends on body condition, about number of larvae, other forms actually, etc. So slow kill strategy, small doses, every day, once per day, once per week, or once per month. It's a different approach, but I think that once per week is the most common strategy. So yeah, I would, with this worm also, I would start with slow kill strategy, but also you need to be aware if you underdose, if you do a small dosage, you maybe are not be able to kill parasite. So I think that during the longer period of time, you need to repeat this therapy and do, like I said, and you need to make one period of 1 week or more after last administration of drug and the first checking. For instance, with Berman, you need to make a gap, and this is the rule for every parasitology. So you need to make at least um, uh, 5 to 7 days of a gap without any drug, because you can have a false negative. You can have in vitro, but difficult to prove in vitro. So repeat diagnostic and repeat treatment if you need, and always look at the animal. If you see that there are clinical signs, just go for it. If it's negative, it's negative, but if you see clinical signs, do diagnostics and repeat it. Of course, once you prove antibodies, you will not use that diagnostic tool anymore, because antibodies, they are going to live for a longer period of time. But antigen, PCR, Berman, yes, to check it. Thank you. It's easy about Dirofilaria and Trypanosoma. In wet blood smear, I would always do some staining, but Dirofilaria is really easy to diagnose, because you have larva, moving larva, and yeah, it's quite big larva, and it's really fast moving, and but I will always do staining, because there are different kind of Dirofilaria. You have imitis, you have rapens. So if you find a larva in blood smear, it's just the beginning of the diagnostics. It's not the end. You need to be sure that is Dirofilaria sp, and yeah, it's easy when you stain. You have on a head part, you have one gap on a larva, one gap without stained parts, and the Dirofilaria repens have um, have all stained, um, all parts of head is stained. So yeah, I will use stained smears. Um, do you have any more photos of larva? Actually, I had one film, and I will try. I will try to share, but it's not part of presentation. That was my something else. Let me see. I'm not sure if you can see this. So this is how Berman techniques look like when you collect from the bottom of the glass. You will see this kind of larva. I'm not sure if you could see. Which stain is used? I'm using. You can use Diff Quick, or I like to use right stain. I will write you. This is stain I'm using. So yeah, Diff Quick or right stain, and this is perfect for blood smears. So I hope you could see this short film with the larva. Oxyuris equi, I need to search for that because horses are not so easy to. They have problems with that. I will tell you what is the perfect. I don't know by heart, so I will tell you uh, on LinkedIn later about it. Yeah. You're welcome. So I'm not sure if I cover all your questions. They are really great. I really enjoyed questions. So if you have any kind of questions from my field, I will tell you um, whenever I can. Okay.
Any other question? I think there is no more question left. You have resolved everything very well.
Okay.
So I would like to end this session. So, dear participants, we extend our heartfelt thanks to Dr. Katarina Novakovic for her invaluable insight during today's webinar. And we also thank all the participants for their active engagement contributing to the success of the event. And thank you, everyone. We appreciate you being here. So, dear participants, I have last announcement for you. I'm going to share a feedback form into the chat section at the end of the session. Please fill out the feedback form so that we can improve our future webinar and will perform more session like this. Thank you so much, Dr. Katerina. We are overwhelmed for your
Thank
time.
you. Thank you. I hope I helped, and yeah, um, enjoy parasitology. Good luck to everyone.
Good luck to you.
Thank you.
Good luck